PhD student who did the first measurements of the aluminium, how much aluminium there was in the brain tissue, came to me with the results. I told her to go back and look at all of this again, because we thought we had seen astronomical amounts of aluminium in familial Alzheimer's disease, and then we saw values at least as high in autism brain tissue. And as I wrote eventually in the publication that we made in the abstract, you know, why a 15-year-old boy brain, when he died, would have amounts of aluminium almost never measured before is almost incomprehensible. So yes, we suddenly found something which I did not expect at all to find. We did the same thing in the 10, looked where the aluminium was in this time in 10 individuals who died with a diagnosis of autism. We found something completely different. We found that virtually all of the aluminium was not in neurons. It was in the housekeeping cells of the brain, which are such things as the glia and the microglia. It was also in lymphocytes, white blood cells, macrophages that appeared to be crossing from the blood into brain tissue, across the meninges, across the blood-brain barrier. We all of a sudden saw that aluminium was potentially getting into brain tissue via white blood cells, via macrophages, via lymphocytes. And you know, this was that moment when all of a sudden, yes, I can now begin to understand why we can find so much aluminium in autism brain tissue. And the reason is because previously there was no evidence that these types of cells were playing any role in putting aluminium into brain tissue in, for example, aged or individuals or Alzheimer's disease. The mechanisms there were still unknown, actually, but there was no evidence that that was the way in which aluminium was getting in. Whereas for these autism brain tissues, it was probably the mechanism whereby aluminium was getting in. And of course, it fitted remarkably in some ways with our research looking at what happens to aluminium at the vaccine injection site.
Right.
Because at the vaccine injection site, where an aluminium adjuvant is in the vaccine, what we saw were these same types of white blood cells, macrophages, filling up with aluminium. They could actually ingest it until the whole of the volume of the cell was full of aluminium without causing them any immediate damage. They can then go off around the body. They can go off to the lymph nodes, perhaps helping to initiate the actual immune response that the vaccine was given for. But they can go elsewhere. They could go to brain tissue. Roman Girardi's group, using rats, showed that they could go from an injection site to brain tissue. So we now have a mechanism for loading up brain tissue with aluminium, and that is through these housekeeping cells of the body.
We're injecting this stuff, so he did work and saw that it is definitely traveling straight to the brain, and therefore the university shut him down and destroyed, you know, the career of the most prolific aluminum scientist in the